Good evening friends. Today we will discuss about indirect Elisa. Before that, I request all of my friends, please subscribe my YouTube channel and also watch, like and share my videos with your friends. Indirect ELISA is a biochemical assay that is commonly used to detect as well as quantify antibody against a specific pathogen. Like other immunoassay, it is also based on highly specific antigen-antibody reaction. In direct ELISA, only one antibody is used for detection, whereas in indirect ELISA, two different antibodies are used. One is a primary antibody that recognizes and binds to the target antigen, and the other is a secondary antibody that binds to the primary antibody. The secondary antibody is linked with an enzyme that converts a chromogenic substrate into a colored product, which is then measured by a plate reader. Indirect ELISA is commonly used to detect antibody against a variety of viruses such as hepatitis A, B, C, human immunodeficiency virus and others. This assay is most frequently used to determine the outcome of an immunological response, such as measuring the concentration of antibody in a sample. Indirect ELISA is typically carried out in 96 wells polystyrene microtiter plate. For example, we want to test the blood sample of a patient to detect antibody against human immunodeficiency virus HIV. First of all, we have to collect the blood sample and then extract serum. First step in indirect ELISA is the immobilization of antigen at the bottom of the well of a microtiter plate. Antigen are immobilized by direct adsorption at the surface. For this, a small amount of solution of purified antigen is coated onto the well and then incubated. So these are the antigens that are immobilized at the bottom of the well. After incubation, unbound antigen are washed off and then the plate is blocked by adding blocking buffer or protein such as bovine serum albumin. Blocking is necessary to ensure that the primary antibody binds only to the target antigen. Friends, here there are only 4 immobilized antigen, and these are the free site or unoccupied site. So when we add the primary antibody, the antibody will bind to the target antigen as well as to all these remaining sites. If the primary antibody will also bind to these sites, then there will be error. So to prevent the non-specific binding of primary antibody, the plate is blocked. For detection of antibody against HIV, purified recombinant HIV proteins such as GP41, GP120 and P24 are used as antigen. Blocking is followed by primary antibody incubation. In this example, patient serum is the source of primary antibody. In this step, a small amount of patient serum is added to the well and then incubated. In place of serum, any other sample suspected to have the primary antibody can also be added. During incubation, primary antibody, if present in serum, will bind to this immobilized antigen, and as a result, antigen-antibody complex will be formed. So these are the primary antibody that binds to the target antigen. The primary antibody will not bind to the remaining site. This is because the sites are already blocked by adding bovine serum albumin. During incubation, some primary antibody will bind to the target antigen and other primary antibody will remain free or unbound. Such free or unbound primary antibody are then washed off from the well by using phosphate buffered saline. Next step is secondary antibody incubation. In this step, a small amount of solution containing secondary antibody is added to the well and then incubated. During incubation, some secondary antibody will bind to the primary antibody and other secondary antibody will remain free or unbound. Such free or unbound secondary antibody are then washed off by using phosphate buffer saline. So these are the secondary antibody that binds to the primary antibody. The secondary antibody are linked with an enzyme. Next step is substrate addition and detection. In this step, a suitable amount of chromogenic substrate is added to the well and then incubated. We know that the secondary antibody is linked with an enzyme. So when a suitable amount of chromogenic substrate is added, it is converted into a color product. After color development, further enzymatic action is prevented by adding a stop solution containing sulfuric acid. The amount of product formed is proportional to the concentration of antibody in the sample, such as serum. The color intensity of each well is then measured by a plate reader. In order to determine the concentration of antibody in the sample, a standard calibration curve is prepared. In order to prepare a standard calibration curve, a series of standards are prepared by taking increasing known concentration of primary antibody in different wells of the same microtiter plate in which the sample has been placed. All these wells contain increasing known concentration of antibody. In this well, we have placed the sample. We want to determine the concentration of antibody present in the sample. All the standards contain increasing known concentration of primary antibody. In next step, remaining reagents are added as per the Elisa protocol. After color development in all these wells containing standards and in the sample, Further enzymatic action is prevented by adding a stop solution containing sulfuric acid. The optical density of all these wells is measured at a wavelength of 450 nanometer by a plate reader. After measurement of optical density, a standard calibration curve is plotted by taking concentration of different standards on x-axis and their optical density or absorbance on y-axis. From the standard calibration curve, the concentration of antibody in the sample, such as serum, is determined by interpolation. In this example, patient serum contains specific antibody against the immobilized HIV proteins. Because of this, during incubation, serum antibody will bind to the HIV proteins. Serum antibody is the primary antibody. After this, when enzyme-linked secondary antibody is added to the well, there will be binding between secondary antibody and primary antibody. Because of the specific binding between target antigen and the primary antibody and between primary and secondary antibody, when the plate is washed by using phosphate buffer saline, this bound antibody will still remain inside the well. In the next step, when the chromogenic substrate is added, colored product is formed. So color development indicates a positive result. That means patient serum contains antibody against human immunodeficiency virus. So we can say that the person is infected by HIV. On the other hand, if serum or any other sample do not contain specific antibody against the target antigen, then there will be no binding. In the absence of primary antibody, when the enzyme-linked secondary antibody is added to the well, all secondary antibody will remain free or unbound. In the next step, when the plate is washed, all secondary antibody will be removed from the well. In the absence of secondary antibody, when a chromogenic substrate is added, colored product is not formed. So absence of color indicates a negative result. That means serum do not contain antibody against HIV. So we can say that the person is not infected by human immunodeficiency virus. Friends, in this technique, the antigen is detected indirectly by using enzyme-linked secondary antibody. Hence, the name is Indirect Elisa. Indirect Elisa has several applications. Number one, this technique is used for the detection of antibody in serum and other biological samples. This allows for the estimation of total antibody. It is also useful for the detection of antigens, toxins, proteins, and glycoproteins. Indirect ELISA is a diagnostic tool in medicine, plant pathology, and food industry. In addition, this technique is used for the early detection of certain cancers and also in the detection of drugs and hormones such as human chorionic gonadotropin. Next, advantages. As compared to direct ELISA, indirect ELISA is more sensitive, flexible and also economical. This assay is more sensitive because multiple level secondary antibody binds to the primary antibody. This amplifies the signal, thereby making this assay more sensitive. Number two, this assay is also flexible since same level secondary antibody binds to different primary antibody. In addition, it is also economical because compared to direct ELISA, fewer level antibodies are required for this technique. However, indirect ELISA has several disadvantages. Number one, in this assay, there is a potential for cross-reactivity. This is due to the binding of secondary antibody with non-specific proteins. In addition, this assay has one additional incubation step, thereby making this technique more complex than direct ELISA. In addition, there is also increased background noise. So friends, this is all about indirect ELISA. For more updates, subscribe my YouTube channel, Sam Biology Class. Goodbye, good night.